started. Hi, everybody. Good afternoon. Thanks for joining us today. My name is Gabi Conacher. I am founder of Wishes for Elliot, and under which Deep Connections is a project. And we're really excited today to continue our series on clinical trials. And I see that this is the old slide. Sorry, it doesn't have a full title, but what every rare should know. But no matter, I just wanted to welcome everybody. We have some fantastic guests with us today, and we hope that you will share your questions and thoughts and concerns. Feel free to go ahead and document those throughout in the Q&A or the chat function, and we'll keep an eye on those. And just a couple of quick housekeeping. We're recording this session and it will be online in a couple days. All the panelists will be on video, but attendees are muted and not on video just to control for sound, but we do really want your engagement. So please stick with us and put your comments in those sections so that we can connect with you and engage with your questions. With that, I think I'm going to go ahead and pass it off to Mery. So basically if people have come to enough of our webinars, they know that we always make a point to include the caregiver voice. And today we're really excited to have Mery to talk a little bit about what is going on in her community and questions she has and how she's kind of preparing for the idea of clinical trials. So.
All right, thank you, Gabby. So my name's Mery Oman. I am mom to Ethan. He just turned 4, and he was born with severe intractable seizures from day 1 that was found to be due to SCN2A mutation. And so I'm also a board member of our foundation, and I'm involved in helping whatever our executive committee needs my help with. Mostly I'm more involved in the research committee, so this is all very relevant to me. So we do have a lot of questions personally and as a community, and as a board member. So our community right now, we are getting ready for trials. We have a couple of therapies that are in the pipeline, and we're hoping that it'll be soon, hopefully next year or within 2. But we don't have an official natural history study. One was conducted in another country, and that has not really been as relevant because of all the data. We have not had access to that. And right now we're working with Ann Berg, Lori's Children's. She is gonna be the PI for our clinical trial readiness study, which will be a prospective longitudinal study over 18 months. And it's gonna measure some endpoints in our kids. And she's hoping that this data will be very relevant for trials. To pharma or whoever will use it to, see what can be done for our kids and what the results are. So as a community, I guess I was telling Gabby that it feels very distant because we've been waiting and waiting for something to happen, like a cure, right? We're waiting for the ASO therapeutics to happen, which they keep telling us it's going to be hopefully this year, hopefully next year. And COVID has made things very challenging, of course. But I think because the thought feels very distant, it's been very challenging for the community to feel engaged in terms of participation and asking questions of what they need to do and how to prepare. So I guess the questions that we have as members of our community would be whether everyone would be part of the trials or not, whether they would be included or excluded. No one really knows that. They do know, the pharma, the groups that are working on the ASO, they do know that the therapeutic is specifically for gain of function phenotypes. So the families who have the gain of function know that that's more targeted towards them versus the loss of function. And in terms of other questions I think you talked about it on part 1 of this, where the trials will happen, is it going to cost, is insurance going to cover it, all those things, that families do wonder. If a drug does help during trials, would they continue taking it? That's another question that most participants in trials will often ask. Other questions are whether we will have access to results of the preclinical studies done, whether it's like animal testing and all that, before enrolling our patients in the phase 1. We'll be the guinea pigs, the first time being in humans. And then the other questions that we have is more like ethical, whether, would these trials be focused on randomized versus not, because our kids are so sick. There's like an ethical component to whether you should have a placebo, even consider having a placebo in a really sick child. So those are some of the questions that people do think about and that come to mind. In terms of, as a board member, being part of our research committee, there's still so much to do as we work towards getting ready. So we're working on this clinical readiness study, but our community is so small that we're still working on possibly creating the centers of excellence, as we've seen with CDKL5 and TSA even. I believe they're everywhere. So we're growing, and we would like some guidance in terms of what to do with that. So would our kids have to travel far to see some specialists, or do their, neurologists, could they do the treatments themselves and be part of trials everywhere in the US or internationally even? Other questions that we have are, I talked about ASO being a gene therapy, and we do know about other drugs, including Neurocrine, creating something for SCN2A, so that's a compound. So if one is part of a trial, like a compound trial, would it be excluded in the gene therapy trial? We don't know. So those are some questions that our community would think about as well. Lots of questions. I think that's a good start for now. I will pass it off to you, and I'm sure I'll have more questions coming up. So.
Thank you so much, Mery. I think kind of there's some value in you having just started us off with questions, because I think that's kind of what our community is struggling with a lot of the time. Everything is very mysterious, and we're hoping that through this series we can help demystify kind of how clinical trials work, how decisions are made, how communities are engaged. I mean, that's definitely the trend more now, is that communities are being very deeply engaged in the process of development, as opposed to, we're like, okay, we have something now to test. And I think Carolyn will talk a little bit more about that. But I'm really excited to welcome our other two panelists as well. We have Steve Roberds, who is the Chief Scientific Officer at the TS Alliance, Tuberous Sclerosis Alliance. He has a PhD in pharmacology and significant experience working on clinical trials from both the pharmaceutical side and the patient advocacy group side. So Steve's going to have some great perspective for us. And we also have Carolyn McMicken, who is Director of Medical Science Liaisons at Neurocrine Biosciences. She's a neuropsychologist with an extensive experience in clinical trainings and clinical trials, and rare epilepsy. So I think we're going to get some really exciting insights today, and hopefully it'll spur some more questions and answer some of Mery's. So Steve, I'm going to pass it off to you.
All right. Thanks, Gabi. Yeah, and thanks, Mery, for the great introduction. I feel like we could just have a conversation around your questions for the rest of the hour. But I just put together a small number of slides that I hope will illustrate just one example from the TS Alliance, and just some of the things that have happened, especially in our earlier time. So that's what I want to talk about. And I think the theme that you'll hear is really the role of the community and the power of the community. And that includes those living with the disease, parents, family members, but also the community of researchers and clinicians. And I hope you can see some examples of how those have interacted to, I think, address a few of the questions that Mery brought up. So just so that you know where I'm coming from in this discussion, I wanted to make sure everybody at least knew a little bit about tuberous sclerosis complex, or TSC. So it's a genetic disorder. It causes tumors to grow in many organs throughout the body, primarily the brain, skin, eyes, kidney, heart, lungs. But the neurological manifestations, so the epilepsy and autism, tend to be the most devastating. About 85% of people with TSC have epilepsy, and 2/3 of those are not well controlled. So there's a high difference, a large difference in how TSC affects individuals across, from person to person. No two people are exactly the same, even identical twins. And I won't go into the genetics of that we think sort of underlies that, but certainly many of the folks with TSC are at the same end of, the severity spectrum as many of the other developmental epileptic encephalopathies, although there are some living with TSC who don't and who haven't experienced epilepsy. And so we're glad for that too. So it's a rare disease, but not super rare, about 1 in 6,000 live births. So we estimate there could be about 50,000 people in the US with TSC. Because it's so variable, sometimes there's a lot of underdiagnosis. So I wanted to show you a timeline that I'll advance through. There's a little bit of animation here, but setting the stage, the TS Alliance was founded in 1974 by 4 moms in Southern California. It was before the internet, so they're sitting around a table, they're communicating by letters and magazine articles and phone calls and all of that. And it took 10 years before the first research grant was funded. So TSC and TS Alliance have been around for a long time. So some of the things that you see that we've done, just keep in mind, we didn't do them in the first 10 or even 20 years of our existence. It took time to get to where we are now. In the late 90s, we finally had the breakthrough of identifying the 2 genes, TSC1 and TSC2. That really led to a boost for the community, both, again, the research and the patient community. And so some of the things that the TS Alliance did was to host a consensus conference on the diagnosis and the current way at that time for managing and treating TSC as best the physicians knew. And in 1999, we had our first national conference. This is for patients, families, living with TSC. And so that really brought people together face to face to share experiences, but also to hear from clinicians, other types of healthcare providers, and researchers on what was going on in TSC. So it was helping inform that community. And in 2001, the TS Alliance hired Kari, who's now our CEO, to really lead the volunteer outreach for the Alliance. And we, under Kari's leadership, she created or facilitated the creation of community alliances. These are the volunteer groups around the country. We have about 36 of them right now. So they're not separate 501c3s. We're the only nonprofit, but they're volunteer branches of us, and we provide a lot of support and education and work very closely with the community. So we grew from 95 volunteers, a list of 95 volunteers when she started in 2001, to over 2,000 volunteers today through that engagement of the community. So then a breakthrough, and this is where we get to the clinical trial. So in 2002, biologists studying fruit flies figured out what the TSC genes did. And they regulate the activity of an enzyme called abbreviated mTOR. And the TOR in mTOR stands for target of rapamycin. Rapamycin was a drug that was already on the market. And so when that connection was made between TSC and this drug that inhibited the pathway that was overactive in TSC, you can see beginning in 2003 in these little colored boxes at the bottom. I guess I'll point with my mouse instead of with my hand. These are clinical studies, clinical trials that began. And so starting right away, doctors started testing whether rapamycin might have an effect on many of the manifestations. So the brain tumors, the kidney tumors, I didn't illustrate lung on here. And the numbers indicate the numbers of participants in each of these studies, right? So you've got very small studies in the beginning, 5 people in this one, 25 here, 36 here, 16 here, 28 here. These were all open label studies, so they were not placebo controlled. That wasn't necessarily, to Mary's point, which was an excellent one about ethics. It was simply that it was an early stage. These weren't intended to necessarily prove definitively that the drug would work. It was more to look for a signal, a sign that it would work, and then the double-blind placebo-controlled trials would be developed. But actually, the blue boxes, these are for the brain tumor known as a SEGA, subependymal giant cell astrocytoma. The green ones illustrate some trials for the kidney tumor shrinkage. The signal was so great in this trial of 28 patients, even though it was open label, the FDA actually approved the use of an analog of rapamycin called everolimus or Afinitor for use for treating these brain tumors in TSC based on these data, because essentially everyone responded. But the FDA required that they go back and then do even more rigorous study with a placebo group, and that's 117 that was done, and of course it was positive as well. So that remains, the approval remains on the market. Similar story in the kidney realm, although they did not approve it until after the study was done. So while all these clinical trials are getting started, the TS Alliance launched their natural history database in 2006. So this is a database that collects clinical data. So it's a little different than what you might call a natural history study, where you have enrolled a certain number of people and you follow them, over time. We do that, but we do it in a retrospective or an observational way. That is, people who are coming in for their clinic visits, they can consent to participate in the database, and then the physicians or healthcare staff can enter their data in our natural history database. And we currently have over 2,200 people enrolled. So I wanted to highlight, again, just a couple more slides, some of the themes about these clinical trials. So I think one of the things that I tried to show from the last slide, as you saw, they tended to start very small. They were open label. It was kind of see if it works. I only took you through 2011 because trials have continued, and there's a lot more I could talk about, but I think the purpose of this discussion is really about the early days, right? And especially those first few clinical trials, and how do you get into it? One thing that we've observed is that, especially in the last 10 years, is that biomarkers or validation studies of some other kind often come first. So I have a couple examples. The top one's in the epilepsy space. So a group of TSC clinics got together, formed a clinical research consortium, and applied for NIH funding to do essentially a biomarker study to look at EEGs at regular intervals. So this wasn't just whatever they needed for sort of standard of care. This was actually very aggressive. Like every 6 weeks, they were having an EEG in the first 6 months of life, and then about every 3 months, and then out until 2 years of age. This enabled us, so this was a really strong, this was what I would call a true natural history study. Everybody got the EEGs at the same ages, very systematically, and we went and this was 40 babies, and followed up to 2 years of age. And what that taught us was that we could see changes in the EEG before the clinical seizures came on. And so that led to, again, an NIH-funded clinical trial that is, enrollment has completed, but the trial is still ongoing, called the PREVENT trial. And this is the first preventative trial for epilepsy in the US using vigabatrin. It's a drug that treats infantile spasms in TSC. And the concept is, can we get it on board? If we get it on board when the EEG starts to change, can we then prevent the clinical epilepsy and the developmental delays that often follow intractable epilepsy? So we hope that that'll be certainly positive. I think there are other data out there that suggest the earlier you treat, the better it would be, but those are not controlled, not placebo, double-blinded studies. And it's led to the STOP2 trial, which is just now open for enrollment, which is very similar to the PREVENT trial, but using rapamycin, right? So now using attacking actually the pathway that's disrupted in TSC, which we think will be beneficial not only for the epilepsy, but also for the tumor growth, whether in the brain or the kidney or the skin, etc. So that's kind of an example of stepwise. And then on the autism side, illustrated in the bottom of this slide, that this is all work by Shefali Justi and her collaborators at Boston Children's and some other sites. They first did a study, again, a very thorough natural history study of a small number of infants with TSC to follow the development of autism, and they tested them with different instruments that were age appropriate as they aged up to 3 years. And they saw, indeed, much like the EEG, they saw that there are early instruments that wouldn't give you a diagnosis of autism, say at age 12 months, but they predict that that person will have autism by the time they're 3 years old. So they set up a pilot study that was really for feasibility, again, not blinded at all, or no placebo, if you will. But they did this intervention study just to see, can we see a signal? Does it seem like the kids are improving versus the baseline data collected back here on this other set of kids? Is the study, because it relies on parent interaction, can the parents do it? Does it work for them? I mean, does the process work for them, right? Is it feasible? And the answer was yes. And so then they got NIH funding for a large trial that's ongoing right now, still enrolling, called the JETS trial, that's testing the efficacy of Jasper on early intervention in children with TSC to prevent that development of autism. So I hope those two examples were kind of helpful from a point of view of illustrating. So to summarize, just some takeaway points that I wanted to make sure that I said, and if I didn't, and if I did say them, to say them again, that really, I think one of the key things that helps set the stage for success here is a real united and collaborative community of the patients, the families, the researchers, and the clinicians. By having those early conferences and consensus conference and national conference, and having volunteer groups, everybody's informed, they're working together, they're on the same page. I think that really helped. Another takeaway is clinical trials are just part of a process. You don't, and Mery talked about this, right? You don't just have a clinical trial and get a drug approved. There are steps to make sure that the trial that you do that is really good and set to answer the question. So often, the first study that might be done is a natural history study, some kind of pilot study, open label study, just to get some confidence that you know what direction you're headed is going to be a good direction. Then a feasibility study, often an open label study, to make sure the trial design works. And again, that you've kind of got a signal there. And then the real pivotal study that would be used for submission in an NDA would be very high standards, so very well controlled, rigorously conducted, yeah, and often takes more time and a lot more money because of that. So, I think in summary, I think having something I didn't specifically address, but I wanted to bring up, that the peer-to-peer advocacy. I talked about how the community was brought together and educated. We've really found that, especially in the PREVENT trial and to some extent in the JETS trial, having community members or parents advocate to other parents, hey, are you aware of this study? There's an opportunity to participate in this. That has definitely contributed to enrollment in these studies. And so it can really be successful along the whole process. So I will end. I know this will be recorded, Gabi, and so share these links however you want. Just, I think, some potential toolkits. One is a pamphlet, and it's also on our webpage, of a basic introduction to clinical trials. If any of you want to copy it and improve it, it's a little old. It's a few years old, so there may be some things that need updated. And the Clinical Trials Transformation Initiative has done a lot around clinical trials recommendations on how to improve them. And some of those things touch directly on engagement of sponsors with patient advocacy groups and patient advocacy groups with sponsors. So I think there's a lot of good stuff to be learned. So I put the second bullet point is very self-serving. I was involved in that project, but I think it came out with some nice recommendations. So that's all I have. Thank you. And from here, I guess we go to Carolyn.
Thanks so much, Steve. Hi, everybody. I'll admit that I asked Steve to go before me because you have great information and a long experience in dedicating yourself and your group to really pushing forward the understanding of TSC and potential treatments for people who are living with it. So I'm going to piggyback a little bit on some of the concepts that Steve shared and give more of the industry perspective. So, you know, some of the challenges that he mentioned in looking at the long history of working in the TSC space is that when a newly identified disorder or a rare disorder becomes a point of interest for a group, there's a lot of unknowns. So, you know, in the beginning, and I know some of the people in attendance here might be part of communities that their child's diagnosis was only recently discovered, or their genetic condition. So the earlier we are in the process of understanding that condition, the less we know. And so there's a lot of work, as you saw in Steve's example, that goes in up front before you can even test any kind of intervention for a specific condition. And another challenge, in fact, with rare disease, as all of you know, is that there's a smaller population available to do that research. So I've already been hearing a lot from families and caregivers that it can be burdensome because there's lots of questionnaires and surveys to fill out, and you want to make sure that by contributing that it's going to be pulled through to the next step. And likewise, when we have a small group of people that we can keep going to try and understand the condition better, as well as potentially test a new intervention, we have to balance out, as always, the safety and looking at what's necessary to really make sure that we can get that treatment available, as well as what's feasible for the community and for the children. And there's a lot of strategies I think that Steve touched on, and I can go into in more depth in terms of overcoming them. So certainly these partnerships, partnering with the rare community and with organizations like Deep Connections and Wishes for Elliot, as well as partnership with different stakeholders, so your clinicians, researchers, the FDA. And I can share a little bit about my experience. So perhaps for another time, we can give some information on the difference between where I sit now in my organization, Neurocrine, which is in medical affairs, and the clinical development space. So I've always in industry sat in this medical affairs space, and we usually come in to support clinical trials, but also mainly once a new treatment becomes available, we support a lot of that later phase research. However, in my kind of past lives, I've worked as a research coordinator on clinical trials and supported several clinical trial programs. And what I have come to appreciate is that from a pharmaceutical perspective, conducting a clinical trial, especially in a rare disease, it requires a strong commitment, and it does require that partnership with patients and families. Another thing is, I think was really well illustrated in the TS Alliance story, is that we know that we're going to potentially have some setbacks, and we are persistent in the face of them. And I appreciate that when there's we all have a sense of urgency, and it can be really frustrating and challenging if there are setbacks, but I always try and keep in mind that the well-being of the patient or the child is top of mind, and we have to balance out that safety as well as the pursuit of something that's going to help. And speaking of safety, whether it's a rare condition or something that's much more prevalent, the same ethical standards apply. So we really need to maintain the security of patient data, and we have to make sure that all of the safeguards are in place to protect the patient, as well as the integrity of the trial. So, you know, especially with rare disease, we don't have a lot of quote unquote shots on goal. So we want to make sure that we carefully plan and do things right the first time. You know, I wanted to kind of connect to some of the experience I have that aligns with Steve's work, because prior to becoming an employee of Neurocrine, I worked for Lundbeck, which some of you might be familiar with, and supported our programs for vigabatrin and infantile spasms. And now there's a compound that has a lot of challenges, safety-wise, as well as because we were trying to help people with a rare disorder. You know, with the babies with infantile spasms, some of the early trials were challenged because of the potential to develop visual field defects, and being able to even monitor that in a baby is nearly impossible. So we had to figure out ways to be safe, but also to continue and get the data that was required. And some trials weren't feasible, especially if you're looking to do a double-blind trial in a vulnerable population. And then another example from prior experience was in the Lennox-Gastaut syndrome space, and working with clobazam or Onfi. And that really exemplifies that we have to get innovative and creative sometimes when it comes to treating a rare disease or designing a trial to treat that rare disease. So in that case, one of the innovations was really exploring that compound over a larger age span. So it's very unusual that you'll see a clinical trial that is ages 2 to 60. And that's what was done in that space in order to get the data that we needed. So there was a question, I think this connects to that question of, you know, is it ethical or safe to have a placebo-controlled trial in a vulnerable population of children with rare epilepsy? And that is a concern. And with the FDA requirements, it really depends on if it's a new chemical entity, a new drug, or something that was already in existence and approved for something different. So when you're talking about a very new treatment, the gold standard and what the FDA requires is to have that placebo control. And what does that mean? So it doesn't mean that that any trial would not treat a group of the children. Typically in a trial of children that have, at least if we're speaking specifically with epilepsy, that are having seizures, then they would be on whatever standard of care medication is available. And the new treatment would be in the active part of the study, it would be added on to whatever other stable medication that the child is on. And we do know that we're not able to keep children just strictly on a placebo and not other medication. So there's other safeguards in place that the FDA requires. I think Steve alluded to this, that there, and I think Mery, you asked a question about, you know, will we have phase 1 data available, the preclinical information. So there is, you know, a whole host of work that's done before you would even test a new treatment in a human being. And it does start with the lab and with animal testing. And oftentimes those results are published in a journal, probably not one that's on your, you know, your dining room table or the coffee table. But but they are typically available upon request. And then there's also a phase in phase 1 where that treatment is tested in healthy subjects, so healthy adults, to make sure that if there's any safety signals that they would pick it up there before testing the medication in the population of interest. And then many of you would become aware of a new treatment potential trial in phase 2. And that's where really it's kind of testing out if the drug at that specific dose is going to show a signal that it would help. So many of those trials are placebo-controlled and feature more than one dose of a particular treatment. And then you would move on to phase 3 if you get positive data in phase 2. And one of the kind of incentives for a rare disease is that if it is a small population, there's some FDA incentives to develop new medications or treatments that would, if you have really strong data, I think in the case that Steve's example of everolimus, that you could have a single study that's of a certain quality and get a medication approved as long as it's safe and efficacious. So I think those are some of the main points that I have. You know, Steve touched on how caregivers play a role, and you know, all of you who are here and those who are going to watch this recording, it's great that you are educating yourselves and connecting with the community, and it really is just continuing to engage with the community, learning as much as possible, staying on top of what new trials are coming available, and talking to your physician about what might fit for your child. And I know that there's a lot of curiosity around, will my child be able to enter into whatever study is coming up next? Some kids will be qualified for certain trials and benefit from them, and others will potentially benefit from the participation of other children in that trial. So it really varies, but if you just keep looking, there's always a different study that you can evaluate might be potentially beneficial. So I'm going to stop there because I'm sure there's lots of questions that we can toss around with this group.
Yeah, thanks so much, Carolyn and Steve. I mean, you covered a lot of ground. I think maybe picking up right where you left off, Carolyn, in terms of eligibility and how that's determined. And I guess, would you, either of you, Steve or Carolyn, talk a little bit about how you determine the inclusion criteria? And I think it's also worth mentioning that even if a child is not involved in the very first stages, right, there are other opportunities to get your child involved, or if there's, promise to get your child onto that drug. But I think that's quite a big question for families, is about, how do I know if and when my kid will be eligible, and how do you determine that?
So I can kick it off. So the inclusion-exclusion criteria it's a balance of making sure that the trial is going to be safe. So for inclusion, you're really defining somewhat narrowly who you're going to test. So, say you have to have a diagnosis of SCN2A. And then there may be some other inclusion criteria. I think Mery mentioned in another trial where it has to be gain of function, not loss of function. So the inclusion really kind of narrow down and try and make it as homogeneous a group as possible, especially in that earlier phase, because again, we don't have a lot of chances with rare diseases, and we want to get a clear signal as to whether this is going to be effective. And then the exclusion criteria, primarily for safety, but also there to ensure that we're getting as kind of homogeneous a group as we can. And then also, there's some other nuances to that we might be aware that one medication might not interact very well with another one. And so you may occasionally see, this treatment is, if they're already on this medication, that they might be excluded from the study. So it does vary though, depending on what the treatment is.
I think that's where patient advocacy groups can really play an important role if the companies and the sponsors will come to them. And that's why, building relationships can be really helpful, because sometimes a company or even the FDA might define inclusion and exclusion criteria in a certain way, either because of past practice, this is the way we've done it, or this is the way the previous trial was done. So some things are not always based. The first draft isn't always ideal, and coming to the patient advocacy group to review those criteria, as well as things like the visit schedule and how many blood draws, and do they have to be anesthetized for MRIs and all kinds of things like that, can really provide important feedback on the feasibility to the sponsor. There was an epilepsy trial in TSC that one of the exclusion criteria was a person couldn't use a rescue medication more than a certain number of times, right, in the trial. But some people with TSC and uncontrolled seizures, they always need. There are a few, that just always need a rescue med. And it's like their seizures never just 2 minutes long, and so those people weren't included. But they didn't discuss it with us, and we didn't have a chance to say why. Why is this important? So it's best to have that conversation if at all possible, and have those relationships so that it has to meet the needs of the FDA and the sponsor, but it also has to meet the needs of the people who are going to participate.
Mm-hmm. Yeah, and I'm going to ask one more thing that's come up before, that we had some emails come in with questions, and then Mery, I want to pass it to you to kind of see if you have some questions that you'd love to get clarified. So kind of bringing a lot of this together. So, with the inclusion exclusion criteria, and then, families also needing to make some tough decisions about weighing the pros and cons for them if they have to come off of drugs to be a part of a trial, or decide that they won't give rescue meds as frequently. I guess, how do you see this push towards more patient-centered outcomes, and really, looking at the people that you're working with as more than just we've got to get rid of the seizures, or and really engaging communities more? I don't know if either of you want to speak to that at all.
Well, I could start on this one, and Carolyn follow up. So I think there's a real opportunity there, because as you alluded to, seizure counts aren't the end all and be all, right? We know that seizures come with lots of risks of anything from falling and being hurt to SUDEP. So we certainly want to control, and we certainly want to eliminate the seizures. But there are many patients and families who are willing to accept that changes in seizure, whether it's phenotypes or time of day, or just the decrease in frequency, or the maybe having a regular pattern, so that they come about once a month. It gives them the confidence and the freedom to kind of go out, if you're on a monthly cycle, to go out those first couple of weeks with pretty good confidence that it's, nothing severe. A seizure is unlikely. Not that it won't happen, but that a seizure is unlikely. So there are added benefits to quality of life, work schedules, school schedules, all of that go beyond the that go beyond the seizure counts themselves, and that's not even including other things that we might think of as comorbidities. So, I mean, in the case of TSC, for example, we know that untreated infantile spasms are highly correlated with developmental delay, irreversible developmental delay. But if we could reasonably control those seizures or decrease their frequency, or maybe we get rid of the infantile spasms, but they still have some focal onset seizures, maybe they don't get the developmental delay, right? That's what some of these studies are going to look at. And so again, the seizure count isn't the only thing. There can be other associated comorbidities that are valuable.
Mm-hmm. Yeah, and I would just kind of add on to that for me, having kind of stepped away from this space for nearly 5 years, I've seen a tremendous shift, even more and more valuing the voice of advocacy and parents and caregivers in really defining what is a meaningful outcome mm-hmm. for my child for this treatment, because it's for our kind of science friends and for the FDA, the gold standard for many years was statistical significance. So if you have like 5 seizures and then it goes down to 2 seizures, is that statistically significant? And now there is, even with the FDA, more of a kind of move towards, we want to know what are patient or caregiver defined outcomes and what's clinically meaningful, because it really is about improving the overall functioning by treating this aspect of a disorder or many aspects of a syndrome.
Mery, did anything come up for you in listening to those conversations? Anything that you'd like to probe more on or feel like you still want some clarity on?
No, I think that was great. Just learning from the journey of TSA, the Alliance. For us, I guess personally and selfishly, the question is, how can we how can we expedite things? Because we need a cure like yesterday, right? And I know all our families would jump onto, we fill out as many surveys as possible. We'll do anything we can. Everyone wants to participate because everyone's eager and it's urgent. It's a call to action to really participate. So what can we do as a foundation besides urging our families to do every survey possible and just participate? How can we expedite things?
So I can give a stab at answering. As we've grown, and the Alliance over the last about 10 years, we've started doing more in the research space than just research grants, right? So you saw it start with the natural history database back in 2006. So that started collecting a set of data that then researchers from academia or industry could access to at least get a global picture of what's going on, but also to identify people who would be suitable for certain clinical trials, right? For inclusion, depending on the inclusion and exclusion criteria. So I think having that sort of things, some sort of natural history data, registry, collecting that meaningful data on how the disease is affecting the patients and the families is important. And then we've started collecting blood samples or tissue samples from people who are also enrolled in the natural history database. So in fact, there's one company that's doing a trial in TSC with a drug because they got some of the blood samples from us, found a marker that they'd seen kind of correlate with working in another disease. And they're like, oh, well, okay, let's try it. Let's see. So you've got the data and you've got the blood. Of course, have researchers, and I think that's the the research grants and the research conferences. And then we also have a preclinical consortium. So even more recently, we put together, working with academics, but also industry partners, a small set of animal models that are really reproducible and reasonably translatable to the clinic. So if you see an effect in the mouse model, we think we can understand why there's a reasonable chance for this to perhaps do something in the clinic in people. And so having all these kinds of sort of resources, I hope it makes it easy for somebody with an idea, whether it's antisense or gene therapy or a small molecule, to say, wow, they've got all these pieces together. They've got the community that's organized. They've had successful clinical studies and trials. Why wouldn't I work in that space? We need to go there and see if this works. So you can't build it all at once. And we're 47 years old, something like that, 46 years old. But each of those pieces, so the more that you put these pieces together, I think the more you can, you're ready for something to happen. As I pointed out, was that link from, in fruit flies, from the TSC genes to the mTOR. And as soon as that happened, the light bulbs went on, and we already had TSC clinics. We already had research conferences. We'd already had a family conference. So things were in place to take advantage of that when it happened.
Great.
I would just.
It's amazing. We'll hopefully get there someday, and
Oh,
I know
yeah.
we will.
Yeah.
Yeah.
I was going to just add, in order for it to be the research to be accelerated, it has to happen within groups that kind of hold that information and are able to share it with whoever thinks they're on the cusp of making a breakthrough. So for example, what Steve has described is, and I've heard others say, it's like a kind of industry term, pre-competitive space. So it's not just one pharmaceutical company that's holding and collecting all of that data. It's in partnership with advocacy groups and the ability to share it broadly.
I might just build on that for a second, and Mery, feel free to jump in again. Maybe you could talk about you highlighted it more marginally about the importance of data, natural history registries, and the burdensome nature of them, right, Carolyn? That the families feel like, oh my gosh, I have to find all my records and I have to put everything in. But how is that a really important thing that families can do? Maybe talking about like what role that, or not role, what need that fills, for pharmaceutical companies looking to build in that space potentially, and what kind of information that provides to you and makes it kind of a bit more of an ideal community to work with.
So, there's a couple of things. One is that it really helps to inform how we design a trial. It helps to inform how we create the inclusion criteria and the exclusion. I think that the point was made earlier that you can design a trial and then if all of the patients that have that condition are excluded because it's just not realistic for what they're what their lives look like, then you're never going to get anywhere. So really having access to, whether it's the natural history data or caregiver surveys, it really helps to eliminate some of the mistakes that you could make without that information. And it's kind of like, at times you have to slow down to speed up. You know, if you rush forward without some of that foundational information, it can certainly lead to delays.
Mhm.
I feel like the problem with natural history studies is the time, how long it takes to gather all that, and just the time frame of measuring all that information over a lifespan so far of a child. Is it possible for pharma to come to a group and try to measure in a condensed NHS, looking at it, picking certain measures in their lifetime versus the whole lifespan?
Well, there's lots of kind of innovative ways to approach studying the natural history of a given condition. And then there's also other ways to kind of approximate and get a sense of what the clinical picture is going to look like over time. So my colleagues, you know, we have a whole health outcomes department. They're much more well-versed than I am in all of this, but you can look into claims databases and identify, you know, one diagnosis and then kind of see how that person moves through their, you know, the health system, and get an idea of what the other diagnoses might be, how long they stay on treatment. So you can get it from that angle as well, without having to wait, you know, a lifetime to just begin the testing. And a lot of this stuff, as we've seen things accelerate, the natural history is to some extent going on along with some of the intervention development. And I think, you know, caregiver surveys and interviews can help to get some clues as well, and better define what are going to be, say, for example, clinically meaningful outcomes for this group of children.
And in our natural history database, for example, is which is why I said it's not necessarily the kind of study that you're that you're talking about, Mery. People can be enrolled at any age, so you do get this cross section, right? So, and then their data can be entered. So if they're enrolled, maybe the first time they go to one of our natural history database clinic sites, maybe the first time they go, they're 4 years old, they can enter all the data from the medical records back, right? Which is one of the advantages of it being completely based on medical records, as opposed to something that people would have to be answering or, you know, doing all the time. The data are there, it just has to be, you know, put into our database. But at the same time, if you get somebody, you know, there are some adults with TSC who are so, maybe they didn't have severe seizures, or they didn't have any seizures. They never knew what these bumps were on their face. The doctors didn't diagnose them correctly. And then they start having kids with TSC, and then they get diagnosed, right? And so then you've got the adult who you're probably not going to have his or her records from when they were 10, you know, 5, 10 years old. But that's okay. You've got records from them, maybe from when they're 30 to 40, and you've got records from their kids now from when they were, you know, born until they were 10. And so, and you put all that together. So it's imperfect, but the amount of the data can still be suitable for, at least for hypothesis generation, right? Looking for patterns, looking for big signals, looking for concepts, answering questions like, okay, what proportion of adults who are seen at the clinics, you know, who participate, have kidney lesions, and how many and how big are they? And you can at least get a good, you know, number from that, even if it's not a perfect data set, like to use as a control group in a clinical trial, right? Because that's one way that some natural history studies can be used. I think the data that were collected in the clinical studies that I talked about, where they enrolled the babies from they were less than 6 months old, followed them to 3 years, those could probably be used as control groups if you had to, because of the rigor of the way the data were collected across the sites. So it's kind of fit. I'll stop rambling, but it depends on the study, the fit for purpose, but you can certainly get good ideas just by piecing it together as you suggested. You don't have to not collect data on somebody because they're already 5 years old. You can start at 5 and follow them.
Yeah, and I think that's a really important point. And a lot of us are trying to play catch up and build what we can to help prepare our communities for clinical trials. And this is a big question, but we're running low on time. I just wanted to quickly just fill it in, because I think we had somebody write a question about kind of how the FDA fits into all of this, and like how they regulate this process, that it's not just, pharma companies saying, this is what we're doing and this is how it's going to go. It's very heavily regulated. I don't know if either of you want to give a little synopsis about how you have to coordinate with and they regulate, communications and all those kinds of things.
Mm-hmm.
Well, I can start. When it comes to developing a new medication or a treatment, it's a partnership with the FDA every step of the way, and they are there to ensure the safety of the patient population that you're studying, as well as to ensure the highest standards possible of the data that you're getting for those individuals. So there are regulations pretty much around everything, including, especially in the kind of pharmaceutical space, how different individuals who are part of, say, my company interact directly with patients or with clinicians. There's another layer related to publicly traded companies in terms of what can be disclosed to the public as well. And we strive to meet all of those standards and still be within those regulations as transparent as possible, in particular when it comes to the safety of patients. So I don't know if that answers satisfactorily, but I'll let Steve add to that.
Not too deep, but I think that's a great kind of overview. Steve, what would you add?
So yeah, I would add that. So that's all true, of course, but the folks at the FDA that I've been interacting with over the past few years, and by that I mean at conferences, at meetings, at workshops, whatever, are really open to new ways of doing things. Okay? The problem is that we kind of need data on, why the new way is going, is better or is going to be feasible. And so there's a lot of, there may be preliminary work that needs to be done, but they're certainly open to ways. There's an interesting example that I didn't know about until I was at a conference, and is in the migraine space. So there's actually guidance for development of migraine medications that is allows two different endpoints, and so migraine is associated with pain, right, headache, but it's also, many people have nausea, they have sensitivity to light, sensitivity to sound. The FDA has developed guidance where you can get approval for a medication using two endpoints. One has to be the pain. In migraine, that's the key point of pain. But not everybody has nausea. Not everybody has light sensitivity. So what you do is when somebody enrolls, they tell you what's their most bothersome symptom besides the pain. And so it helps with the data because you're not, if you were trying to look at all 4 of those things, part of the people are already, they can't get any better. They don't have light sensitivity. How's the drug going to help, right?
Yeah.
So they developed the process to say, okay, you tell us what your most bothersome symptom is, and then that's what we're going to measure, you know, at the end, in addition to the pain. So it's those kinds of they're open to ways of thinking that are novel, but are still meaningful and rigorous and important to patients. So I think there's a lot of opportunity to work with them. I think that's the really good news.
That's great. Well, I know we're really a little bit over. Do you guys have 1 more minute to stick around to answer 1 more question?
Sure.
So another question that came in, that I was actually speaking with a mother who has a child who's very severely affected by their disorder. And she had concerns that, you know, because her child was very severely affected, that he was unlikely to be part of clinical trials. And I don't know if you want to talk, if you can share some thoughts on why they may or may not, you know, be able to be enrolled early on in trials, and what are the considerations for why they may not?
So, you know, just as a disclaimer, right, every study is different. But oftentimes there's kind of a general principle that anybody enrolled into a clinical trial has to have a certain defined level of medical stability. Now, in kind of rare developmental epileptic encephalopathies, that definition of stability will likely be different than in a different type of group of people. But certainly, again, it's about safety. And, you know, once a medication gets to a certain point in the kind of research process, even if a child isn't qualified to participate in a study, there are potentially other avenues for obtaining treatment. And again, it's really always in kind of discussion with their physician and really kind of understanding what potentially could help or hurt. So that's, you know, and it's not a satisfactory answer because I have heard that question. And, you know, when you feel like you've exhausted other avenues, it's not easy to sit around and wait for others to participate in the trial.
Right. Yeah, there are advantages though that you're, you know, protecting your child from any unforeseen early, you know, things that they discover early on in these trials. Save your child from that. But it is, as speaking as a mother of a child with refractory epilepsy who is severely affected, yes, we want something now, but sometimes it's worth the wait. So any final thoughts before we close out? This has been really fantastic. We of course have lots more questions, but we're not going to get them to them today, and we'll continue the conversation. But any final thoughts about takeaways? I mean, Steve, you gave some great ones. Mery or Carolyn, anything to add?
I think the data is so important. Data is the golden ticket to get us to trials. And I was just thinking, I mean, it's we know our data, our groups, our kids, we know everything. And if there's a way for us to, you know, bring the data together in a safe way, you know, HIPAA compliant. I don't know if there's such a thing with like organizations doing your own data, but some might not have the opportunity to bring a PI and just the time and money involved in gathering that data and analyzing it. If there's just a way for us to do it in a way that would be readily available and efficient, and just to present that to get things going. I think that's the sense here, the urgency again, bringing that up. So I don't know, someday we'll come up with something.
Yeah, well, I think the transition to kind of proving that parent, you know, contributed data has a lot of value is a huge step forward. And I'm hopeful that we're paving the way so that as more rare diseases come forward, they're not going to have to recreate the wheel for all of this. I think that hopefully we're working towards that, and with incredible partners like, TSA, who are incredibly giving and sharing of their experiences and tools and knowledge, is only going to move us forward faster. So really grateful to all of you for taking time today to be with us, our guests, as well as all of you who have joined us. So I'm going to pull this up just to officially quickly thank. So our guests, Carolyn, Mery, and Steve, of course, from Neurocrine Families SCN2A and Tuberous Sclerosis Alliance for joining us. And we have a couple other webinars coming up towards the end of the month. One is on VNS and other brain stimulators to understand if there's something that might be worth considering for your child. And on the 28th, part 2 of our SUDEP series on part 1 was, what do we know? What does the research say? And part 2 is, what can we do? So we're really looking forward to seeing hopefully a lot of you there. And as always, be in touch if you have questions. And this webinar will be up in a few days. So thanks, everybody. Have a great afternoon.
Thanks.
Bye.
Bye.